And now I'll come on to talk to the second part of the talk, which is more looking into the microbiome and its effect on neurodevelopmental disorders. So to give a brief rundown of some of the types of the difficulties I see within the autism population, autism is a very broad term used, but towards the more severe end in terms of non-verbal, pre-verbal children have a lot more comorbid biological difficulties, including the gut, restricted gut biodiversity and dysbiosis, which has been well published and defined, low Prevotella, low bifidobacteria, and an prevalence of more clostridial species. And the question is, why? Is it because these children have more restricted diets, or is there something else at play regarding how those microbiome populations are seeded? Is there a genetic predisposition, or are these children more susceptible to environmental factors? But no doubt, and again, it's been well published, that children with more severe and autistic spectrum have a reduced gut biodiversity, to gastrointestinal difficulties, bloating, wind, and partially absorbing food, reducing leading to mineral deficiencies, which I explained earlier. Cerebral palsy. So in terms of children who've had brain damage, again, it's that bidirectional feedback between the gut and the brain. Invariably, these children all have problems with gut dysmotility. They have quite a brittle, fragile immune system, leading to lots of courses of antibiotics. So their microbiome is periodically wiped out. Lots of other medications, polypharmacy, leading to an increased prevalence of candida. So the candidal and the microbiome, how that influences the microbiome, and not only with motility, but with susceptibility to seizure, the vagal feedback we know, which is well published now, looking at how gut health, when gut issues are worse, seizures tend to get worse as well. Looking at other specific syndromes, such as Down syndrome, where again, dysmotility is very prevalent, along with reduced biodiversity of the gut microbiota. The effect of the microbiome on anxiety and mental health. So the relationship of serotonin production, melatonin production, and other neuropeptides. The impact of populations on seizures generally, I think, is quite misunderstood at the moment, but certainly data ripe for capture in trying to understand children with seizure disorders and how that influences the underlying microbiome. But through all these populations of children with different conditions, I would say these are the points that really unify them are the GI issues, the sleep disturbance, seizures, pain, and sensory processing problems, the ability to integrate and modulate the senses we all have in the world around us. So whether that's hypersensitivities to noise, to sound, to touch, to light, or an inability or hyposensitivity to proprioception, to vestibular sense. And then these are the kind of the main, in terms of defining roles of the microbiome in pathogenicity, which I'd like to talk about. So one is brain architecture. The second is molecule production, such as glucocorticoids, steroids, sex steroids, neuropeptides, and monoamine synthesis. The training of the immune system, which is a really interesting topic, and something I'm really, I think, is kind of the future of this area. The motility, so vagal feedback, and the dysautonomia, which I described. Gut barrier function, so how that is relevant, and there is a lot of similarities between gut barrier function to the blood-brain barrier, and how the microbiome can influence gene expression, epigenicity, and then also the metabolic byproducts, and thinking about with restricted gut bacteria, does a large clostridial population leave you with a buildup of unwanted metabolic byproducts, which potentially could be neurotoxic? And again, certainly in mouse models, that's been evidenced with fulcresol and propionic acid. So first of all, in terms of brain development, so different microbiome populations can really influence certain parts of the brain. So with the hippocampus, looking at serotonergic expression, neurogenesis, BDNF, the hypothalamus, so looking at social hormones such as oxytocin, and the pituitary, so adrenal gland hormones, and sex differentiation, the prefrontal cortex, so executive functioning, myelination and mRNA expression, and the amygdala, so leading to differences in subregional volume and cell morphology. The other thing is looking at glucocorticoid sex steroid production and the differences seen between different microbiota populations. So reduction or changes in cortisol, sex steroids such as testosterone, estradiol, progesterone, and neuropeptides such as oxytocin and vasopressin, which are all really important. They're pro-social and can really be affected in conditions such as autism. And as we discussed, serotonin and dopamine. So serotonin dopamine imbalances are very common in many developmental conditions, including those with anxiety, ADHD, autism, which can have a profound effect not only on behavior, but on sleep, and really impact long term. And then training the immune system through gut permeability. So dysbiosis in general, also dysregulated bowel flora, can really impact on what is let through. So that IgA, gut mucosal barrier, and if specifically the timing is very important. So within the first year of life, if the gut is allowing in things it shouldn't, then children are more likely to have a whole host of other different problems. And I've no doubt this has a profound impact on brain development and the way the immune system is trained, because antigens get through which shouldn't, and therefore they're recognized by our immune system, predisposing more to things like autoimmunity, but also the microglial monocyte activation and how, and the thing to realize is, again, that the bidirectional relationship between the gut and the brain, and how cells within the gut are so important in the training of not only the local immune system, but also potentially of brain monocytes such as microglia and dendrite formation. Dysmotility, so vagal feedback. So as we alluded to in cerebral palsy, with a problem with innervation to the gut and the feedback to the brain, there can be a real problem with motility, but also dysautonomia. So a really strong response to stress and an inability to control internal homeostasis, such as heart rate trajectory, fluctuations in other parameters. And then lastly, in terms of restricted biodiversity and the imbalance there, so big things that we have a big impact on developmental trajectories are things like exposure to antibiotics. So both in utero and postnatally, I don't think we truly appreciate the impact of a course of a 5-day course of IV Ben-Pen Gent early on has on brain development. If you're completely wiping out gut microbiome, you are potentially completely changing that individual's not only microbiome population, but the immune system and brain development, which is a really challenging area to try and get your head around, because as pediatricians, we always try and worry about things like infection early on in children, and especially in preterm children, but invariably prolonged courses of antibiotics, especially within the first year of life, could have a big impact on how that child's brain develops. The impact that high-fat diets have, the westernized diet, the impact of acute systemic inflammation, again, on the biodiversity within the gut. And there's within conditions such as autism, there are different schools of thought regarding pathophysiology, and one is maternal immune activation. So as I alluded to earlier, with the increasing incidence of autoimmune disorders and the impact that can have on offspring with neurodevelopmental disorders. So many papers have demonstrated that mothers with poorly controlled autoimmune disease have a far higher rate of conditions such as autism, especially quite severe aggressive autism, and how that is linked to the microbiome and brain development. There's prenatal autoimmunity. So autoimmunity actually is very, very important in early pruning, and there are certain genes and hyperactivity which can cause autoimmune synaptopathy, and I've given you some examples of those genes there. And then postnatal immune dysregulation, so inflammatory insults from infection and cytokine production can really impact on microglial activation and proliferation. And then gut barrier function and relevance to the blood-brain barrier. So I recently published this in a magazine called Autism Eye, and it was in a study by a Cambridge research lab looking at the role of IgA plasma cells and the impact on not only gut mucosal permeability, but also the protective function it has on the blood-brain barrier. And the plasma cells, which had originally been in the bowel, can migrate, and the molecules, the IgA molecules from those, can actually play a big part in the role in blood in protection of the brain. So again, it reinforces the idea of the gut and the brain being intrinsically linked. And of course, if you're giving antibiotics to wipe these bacteria out, the impact that could have on that immune surveillance and protection. So thinking about the future, really, and I suppose this is my last thing I'd like to share, is thinking about how we could potentially modulate the microbiome and what's been done in the past. So thinking of probiotics, so there have been papers which have shown in mouse models, certainly in Shank, which is a pervasive neurodevelopmental disorder with autistic traits and seizures, that L-Reuteri can impact upon the behavioral pathway within mice. Prebiotics such as inulin, so how that impacts as a fuel on the microbiome and the diversity of it, and how in children with poor diets could have improved outcomes with improving the prebiotics. And then really exciting is FMT, so fecal matter transplant, which is used more widely in the adult population, but may well have an impact in children at risk of neurodevelopmental problems. I know at the moment the Quadrum is doing a phase 1 in ME. I'll be really interested to see the reports on that. So ME generally has a lot of overlap with neurodevelopmental disorders. So that's potentially by repopulating the gut can have a big impact on both biodiversity and all the other issues that we've discussed before. And then there's another phase 2 trial going on at the moment in the US by Jim Adams in Arizona, which is looking at capsular microbiota in autism, again, in children with reduced biodiversity, to see if following antibiotic use, a 2-week course of antibiotics, that repopulating with a more variety of different microbiota, including Prevotella and bifidobacteria, does that influence not only physical symptoms of pain from gastrointestinal difficulties, but does that have an impact on core autism symptoms? Certainly from following on from the initial phase 1, that would seem to be the case, but still early on in understanding and better defining this technology. So that's a brief run through regarding the kind of microbiome and its impact on neurodevelopmental disorders. I'm afraid it was such a short talk, I didn't do the topic justice, but it's certainly an incredibly exciting new area of medicine, which I'm hopeful I've stimulated your interest. And if you are interested in finding further about our mission with the Synapse Center, I'd encourage you to go to the website, which is listed on this slide. But it's a pleasure speaking to you all today, and hopefully you found the talk interesting.